This past January, I went up to the University of Wisconsin to participate in a Tiny Earth workshop where they train instructors of biology, microbiology labs to become their partner instructors, where these partner instructors can go out and teach the Tiny Earth curriculum at their own schools and using their own parameters. So what is Tiny Earth about? Tiny Earth is about student sourcing antibiotic production. Why antibiotics? Well, there is increasing resistance to our known antibiotics among bacteria. We're having more and more problems with bacteria that infect livestock and bacteria that infect people becoming resistant to our antibiotics. We have an antibiotic pipeline problem where biotech companies are not making new antibiotics. They're not investing in research to find antibiotics. It's been 30 years since a new antibiotic has been discovered. Part of this is, the companies, it's money. Antibiotics aren't a big money maker. They'd rather make medications that someone is on all the time because of a chronic disease versus a medication that someone's on for only 2 weeks. So our main need is this finding novel antibiotics. Tiny Earth is trying to find them from soil. Why soil? Soil is an amazingly diverse community of bacteria. Not only is there a huge community of bacteria living in our soil, the majority of our antibiotics have come from soil bacteria. If you think about Bacillus is the main genus of bacteria where our antibiotics come from. But there's a problem with soil. Soil is starting to have problems where our topsoil is eroding, it's being washed away due to climate, more severe climate, weather patterns, and our farming practices. So we really need to think about protecting our soil and analyzing that microbiome that is in our soil right now so we can take advantage of it before this diverse community of organisms is lost. So bacteria, they have metabolites. They have primary and secondary metabolites. Their primary metabolites run their normal systems, photosynthesis, energy production through cellular respiration, DNA replication, transcription, translation, production of any fats, amino acids that they need. However, there are environmental stressors that can throw off bacteria's metabolism, and they can start to produce secondary metabolites. So things like light intensity, pH, nitrogen source, carbon sources, iron levels, and interspecies communication can stress a bacteria. And this is going to cause it to potentially turn its genes for secondary metabolites on and start excreting these natural products. And some of these natural products can be used for growth inhibition, modulation of gene expression, stimulation of sporulation, signal and class diversification, and biofilm formation. But it's these growth inhibitors, it's these secondary metabolites that are growth inhibitors are what we're going to take advantage of because they are our antibiotics. So the first part of Tiny Earth is to go out and get some soil samples. So we were able to bring soil from anywhere we wanted. Particularly, we were told not to bring it from where animals may have been defecating or humans defecating. We wanted to minimize any pathogenic bacteria, but pretty much we could bring back a soil sample from anywhere. We resuspended the soil in sterile water. We did a serial dilution, and then we diluted it out on a media of choice. Usually, and they told us we could pick different medias. We could think about where did our soil sample come from? What do we think it was a rich soil? Do we think it was a nutrient depleted soil? And perhaps that original soil condition might influence the bacteria that would grow. And so we were able to choose different plates and plate out our dilutions. And you can see here's our example of, our dilution plates. Lots of bacteria are growing up. We grew the plates at room temperature. Room temperature, maximum 28 degrees Celsius. If we don't grow them at the higher temperatures, if we avoid growing them at 37 degrees, we're not going to be selecting for pathogenic bacteria, and that's really key for when we start working with these the bacteria from these soil samples, is we want to minimize that risk of having a pathogenic bacteria. And one of the things they told me up at Tiny Earth was they had never cultured a pathogenic bacteria in the history so far. So they felt that the risk was really low to do this in a high school laboratory. Then from those dilution plates, we would pick colonies and create a master plate. So we created a grid on the back of a plate and just plated out as many colonies as we could onto that one master plate. Again, we let it grow overnight at 28 degrees, and now we had individual colonies grown up into a larger group, larger mass that we could go back and start picking from to run a variety of tests on. From that master plate, we then wanted to check which colonies might be antibiotic producing colonies. And to get the bacteria to produce antibiotics, we had to challenge them, start trying to grow them with another bacteria, because once you have these competing bacteria trying to grow, they might start signaling one another, and part of that signaling response could be the production of an antibiotic, a growth inhibitor to the other bacteria. So Tiny Earth has a selection of safe strains that they call their escape strains. And these escape strains are just a series of bacteria that are a good first test to see if your bacteria of interest produces antibiotics. Some of these escape strains are gram-positive, some are gram-negative, and that way you can see, is your colony producing antibiotic against a gram-positive bacteria? Is it producing against a gram-negative bacteria? Maybe it's producing against both types, and it could be a broad spectrum antibiotic. So we would then spread out the escape strain, and then we would pick colonies from our master plate and patch them onto this new plate. And again, we kept a grid, we kept everything numbered, so we knew exactly which colonies were going on which plates. And again, grew them up overnight at 28 degrees, and we would look for the zone of inhibition around our colonies of interest against the escape pathogen. And if you look, it might be hard to see, but there are definitely, where the red arrows are, there are zones of inhibition from the picked colony against the escape pathogen that was plated on the whole plate. Then we could further characterize these bacteria that were antibiotic producers. We wanted to try to identify these bacteria. Is it already a known bacteria that produces a known antibiotic? Or is it a known bacteria that's producing a novel antibiotic? Or is it a novel bacteria producing a novel antibiotic? And so at our level, at different, depending on where you are and how much time you have, you can do different levels of classification. We started with biochemical classification. So we did a gram stain to try to determine the shape of the bacteria and was it gram positive, gram negative. We then did another assortment of biochemical testing. We used different plates. We used blood agar plates. We used McConkey plates to try to understand what type of bacteria we were growing. We looked at motility. Do you have a swimmer? Do you have a twister? How motile is your bacteria? And there's a whole variety of media that you can culture your bacteria on to determine if they're motile or not. You can see with the two plates that we have this bacteria. It's growing very diffuse. It was a motile bacteria and it was able to swim away from where the initial inoculation was. Whereas the second plate down here, you don't see that diffuse growth. You see a nice little colony in the middle of the plate. It does not have the ability to swim. And then here is another set of motility testing where we were using stabs, and if the bacteria grew right along the stab, it was not super motile, it was not a swimmer, but if it grew out in a diffuse pattern, then the bacteria were able to swim through this media. And so we would take note of all the different motility tests for each colony that we were testing. We could also perform single colony 16S rRNA gene PCR amplification and purchase the primers, use a PCR amplification kit, perform the PCR amplification, run it out on gel electrophoresis, and look for that amplified gene band. Once we had we saw that we had a product in our PCR reaction, you can then send that PCR reaction out for sequencing. tinyEarth has a list of the primers that you can use for the PCR amplification. They have a list of different companies that give pretty good rates on the sequencing, and this might allow you to actually identify your genus and species of the bacteria depending on how clean your PCR was, and then you can compare all those biochemical tests, the results of all the biochemical tests, to your sequence analysis and ask, did I identify the right bacteria? Do my biochemical testings agree with this DNA sequencing? The next step that you can also do is a crude purification of your antibiotic. And this was amazingly simple, where you would just plate out a whole plate of your colony that produces antibiotic. You would plate out the escape strain that it produces antibiotic against. Then you would plate out your bacteria of interest. So it's being challenged. It's going to produce, you let it grow overnight, 28 degrees. It's going to produce lots of antibiotic. It's going to be secreted into the plate. The next day you come in, you take that plate, that agar, and you chop it all up, and you do an extraction using ethyl acetate or methanol. You just pretty much chop it all up, stick it in a sonicator. You're extracting the antibiotic out of the agar, and then you filter it off, and then you concentrate your antibiotic down. Once you concentrate your antibiotic, you can then test it. You can test it against different bacteria. Again, you can use all those escape strains and see if this antibiotic does have antibiotic properties against each of those escape strains. You can take your antibiotic and test it against other eukaryotic testing systems to try to understand specifically what pathway this antibiotic is interrupting. And again, to try to understand, maybe what class of antibiotic that you have been able to isolate. Once you've gone through this whole procedure, or you don't even have to go through the procedure, you can stop at any point. You can find that you have antibiotic producers, but you don't have time to do any of the biochemical analysis or the PCR sequencing analysis. You can then take your bacteria and put them in a stab and send the stab up to tinyEarth. And tinyEarth will then store your bacteria that produces an antibiotic until someone in their group has time to further analyze it. And so if you choose, or even if you go through the process of analyzing it, but you still don't know what the antibiotic is that's being produced, and maybe it's a novel antibiotic, maybe it's not, but if you send this up to tinyEarth, they will continue to test it and to figure out what type of antibiotic it is it a novel antibiotic? But you can enter all your information into the tinyEarth database. So there's a way to get a login. You log in, you enter all the descriptions that where did your soil come from, what type of soil was it, what media did you grow your bacteria up on, what did it produce, which escape strain did it produce antibiotics against, etc., etc. You put all that information that you've been collecting into the tinyEarth database, whatever amount you have, and then you send them up your bacteria, and they will eventually process it, and they will try to understand. It takes about 10,000 potential antibiotic hits to discover one new antibiotic. So they really want people to send them antibiotics and bacteria that produce antibiotics from all over the world to try to increase the rate of them finding a new antibiotic. I feel like it's a great opportunity to have students learn discovery. They don't know what's going to happen. They don't know what they're going to find in the soil. And it's really exciting and fun to come in and see what grew, what color were my colonies. You end up getting all different color and morphology of your colonies from the soil bacteria. Are they going to produce antibiotics? It's really exciting to see if they produce antibiotics. And then if you can do the classification, it's a great opportunity to try to figure out that mystery. What do I have? And then if you wanted to do research like questions, you can say, well, what happens if I grow it on a different media? What happens if I test it against, if I purify my antibiotic and I test it against fast plant seed germination? What happens to the germination? So there's ways that you can keep extending this project on your own beyond just that initial culturing bacteria from soil. If you have any questions, feel free to contact me. It was a fun workshop, and all costs are covered if you are accepted in.